All right, boys and girls, video number 2. This one is over mitosis. So I liked this picture. There's a lot of extra information underneath each picture, but I'm going to try to summarize it for you. So remember that the steps of mitosis are actually PMAT. They're not IPMAT. Because IPMAT is actually the cell cycle. So remember when I put this on the board that 90% of a cell's life is right here, and that's interphase. Okay, so that is interphase. And then right in here is mitosis, which is prophase, metaphase, anaphase, telophase. So most of the cell's life obviously is in here. That's when the organelles are made. That's when the cell does its job, okay? And then it prepares for cell division, which is mitosis. And by doing that, the DNA replicates, which means copies. And the cell grows, obviously, and organelles are made. But that's all in this right here, which is interphase. Okay, so let's take a look at these drawings. Hang on. So what we have here is actually interphase. Okay, so that's when the cell is just in its regular life cycle, but it does make have the DNA is duplicated. So the chromosomes, which is in the DNA, are duplicated. So this is interphase. This is prophase. Prophase, if you remember from the hand movements that we did, prophase is where the chromosomes become visible. So when you see a nucleus where you can see the chromosomes, notice here it's just a jiggled up mess, okay? You can't see the chromosomes there. You can clearly see the chromosomes here, so that tells you that that is prophase. Now, if you'll remember on the poster, if you have me in class, if you remember on the poster that I covered up some of the phases because this says early prophase and this says late prophase, and we just take prophase as one phase because you're in 7th grade. But what this does do is it shows you a close-up of these chromosomes. So this is a chromosome, okay, and this right here is a centromere. A centromere. It is in the center. And it connects the chromosome in the middle. And it is a place where the spindle fibers connect or hang on to the chromosome. So when it splits, these things separately are called chromatids. So you have chromosomes, which is this big thing. In the center is the centromere, and when it pulls apart, it is a chromatid, okay? So you have chromosome, centromere, chromatid. So here is metaphase. Remember, for the IPMAT, we are now at metaphase, okay? And metaphase, again, remember those hand movements that we did, is where the chromosomes line up in the middle, okay? And that is metaphase. So you can clearly see that the chromosomes are lining up in the middle. What's also happening is these spindle fibers are actually connecting at the centromere. And that's what the centromeres are for. It connects the chromatids together to make chromosomes, but it also gives a place for these spindle fibers to attach, okay? So these spindle fibers come in and they attach themselves to the centromeres, okay? All right, let me close this down. Okay, now we have anaphase. And again, we combine anaphase. These are both anaphase. You can kind of see a little bit of the pinching here, but it's still anaphase. And you can see where these chromosomes are now getting pulled apart at the centromere, because that's where the spindle fiber is attached, and they're pulling them apart. Okay? So again, remember those hand motions that we did. They're getting pulled apart. And these babies now are called, like I said before, chromatids. So a chromatid is a half of a chromosome. Okay, so that is anaphase. Anaphase. And that is obviously the A in ip-mat, anaphase. And see, it says early anaphase, late anaphase, and we look at them together. Then we've got telophase. Telophase is where it's clearly pinching off. The nuclear membrane has now formed around these new chromosomes that have come together or been pulled apart and duplicated. And it starts to pinch, and that is called telophase, because it is preparing for these daughter cells to completely separate. And when they completely separate, that is called cytokinesis. Okay? So, so this is that, when we did ip-mat, we always had the C over here. So it is cytokinesis, and that is cell movement. Kinesiology is the study of movement of your body, and then cyto means cell. So cell movement or complete cell division. So now you've got all of the stages. We have, again, interphase. Interphase, where the cell spends most of its time. Prophase is where these chromosomes become visible. Metaphase, where the chromosomes line up in the middle and these spindle fibers attach at the centromere. Anaphase, where the chromosomes pull apart, and these are called chromatids. Telophase, where the cell starts to pinch, the nuclear membrane is formed. And then cytokinesis, where they are two identical daughter cells. You should be able to look at a diagram and say what phase it's in. And you should also be able to draw the phases if you had to. So if the question was, draw the phases of mitosis, you should be able to do that. And all that requires is drawing a cell. So you have the first cell, and then you have this is just all a jumbled mess. That would be interphase. Prophase. Remember, when we talk about the division, we're talking about the division of the nucleus at first, okay? So prophase is where you can see these chromosomes becoming visible. Then you have metaphase, where the chromosomes are lining up in the middle. I'm just going to do this fast. Lining up in the middle. Anaphase is where, let me erase that. Where the chromosomes are pulling apart. I'm just doing this fast. And then telophase, that's anaphase. And then telophase is when it starts to pinch. The nuclear membrane is visible. You have the chromosomes in there. And then cytokinesis is two daughter cells. So I think that's it for mitosis. I will certainly look over the review and make sure I've covered everything at the end.